Okay. Welcome everyone for this Neuro'binar of December. Just a quick reminder of our session. See the slide. Voilà. So this is a 1-hour conference, and we will begin with 2 PhD students, which have made, they will present each other's work. And after, we will go with 20 minutes of presentation from a researcher. So for this session, we have Maxime Cazorla, sorry. He will talk about a functional photoinducible tool to manipulate brain connections. And the 2 PhD students are Mostafa Seblani and Guillaume Frébourg. So Mostafa will talk about the thesis of Guillaume Frébourg about structural-vascular human spinal cord characterization. And Guillaume will talk about Mostafa thesis about new targets to reduce edema and improve sensorimotor recovery after spinal lesion. So here we go. Maybe, oh, sorry, my Mostafa, you can begin.
Thank you. So I have to wait for Guillaume or no?
No, maybe you can go and it will go just after you finish.
Okay. So can you see my screen?
Yep.
Okay. So hello, everyone. I'm going to present to you a part of Guillaume Frébourg thesis about the structural vascular characterization of the spinal cord, and more precisely on the use of the DCE MRI for the spinal cord. So the international project between Marseille and Montreal in biomechanics and MRI is focused on 2 pathologies. The first one is degenerative cervical myelopathy, caused by arthrosic changes related to aging, which leads to chronic compression on the spinal cord. The second one is the centromedullary syndrome or central cord syndrome, which is an incomplete injury of the spinal cord caused by the cervical hyperextension. So the question, the research question is to determine if it's possible to establish a link between the mechanical causes of these pathologies and the structural vascular consequences in order to better predict the evolution of these 2 pathologies. So at CRMBM in Marseille, the main objectives are firstly to describe the structural vascular alterations, then to identify the prognostic factors. Whereas at the École de Technologie Supérieure de Montréal, the objective is to study within the tissues the causes of these 2 pathologies. So for the MRI part of the project, the MRI part focuses on the vascularization of the spinal cord, which allows the supply of the organ by the nutrients and oxygen necessary to its metabolism. So the perfusion techniques, the perfusion MRI techniques have been already tested on spinal cord and have been proven to be feasible. So the DSC MRI have been shown good results, but it's so sensitive to B0 fluctuations caused by the patient respiration. That's why they turned to DCE MRI perfusion, dynamic contrast enhanced perfusion, T-weighted radial sequence, which is less impacted by fluctuation of the B0 field. So that allows to evaluate the blood volumes and temporal data It allows to measure and estimate the blood flow irrigating the organ, and to evaluate the microcirculatory. So by microcirculation, we mean arterioles, venules, and capillaries. And to do so, they use an extragenous contrast agent, gadolinium, which varies the relaxation time by its passage in the explored tissue. So there are many sequences proposed by manufacturers, but they all present a poor and low temporal resolution, around 10 seconds. But this sequence, the CRASP sequence, have a good spatial resolution below 1 millimeter squared and a good temporal resolution below 2 seconds. So before analyzing the perfusion of the spinal cord after the injection of gadolinium, a pipeline was set up in order to for fast processing. So there are filtering of data, firstly for to extract the spatial noise due to the pure noise, then to extract dynamic artifacts due to the reconstruction of the acquired image, and finally the filtering was constrained to STD average in order to conserve and not corrupt the gadolinium signal to obtain the final image. So here on the left, you can see an axial slice for the spinal cord. And on the right, you can see the signal curve of different regions of interest. So the gray matter, white matter, and anterior spine artery. And you can see the passage of the molecule. So for the post-processing, the post-processing allows us to extract in the filtered image the average signal in the region of interest in order to calculate some semi-quantitative parameters visible here on the left in the graph. So we have the BAT, or bolus arrival time, then the slope, the maximum slope between the BAT and the TTP, which represents the wash-in of the molecule. And we have the AUC, or the area under curve. So the extraction of these semi-quantitative parameters allows us to evaluate the perfusion after spinal cord, along the spinal cord. So here we can see that in the gray matter and the white matter, the semi-quantitative parameter seems not to be so correlated with the compression area. However, for the anterior spinal artery, we have a good correlation with the semi-quantitative parameters and the compression area. So finally, it's possible to obtain maps of semi-quantitative parameters, voxel by voxel. So here we have 3 slices, the above the compression, on the compression, and under the compression, with the relatives DCE images and the relative semi-quantitative parameters maps for AUC, wash and slope, and TTP. So these maps have to be interpreted with caution. And now the parameters, the validation of these parameters is in progress by acquisition on more patients and by switching to 70 MRI in order to increase the spatial resolution. And many thanks for your attention. So if you have a question, I think we have to wait for you. Okay.
Thanks a lot, Mostafa. So Guillaume, you can begin your presentation, and the questions will be answered later after the Guillaume talk.
So you can see the right screen?
No.
No. Okay, sorry.
Yes. Okay, it's okay.
Sorry, I have to move it.
I think it was okay.
Yeah, because the screen were inverted, so I have to move it. Okay. Is it okay?
Yeah.
Okay, perfect. So hello everyone. I will present for you part of Mostafa Seblani's PhD project. He's working on a pharmacological approach to identify new targets in order to reduce early edema, sensorimotor recovery following spinal cord injury. After the direct impact of the mechanical trauma called primary injury, a secondary injury will occur. It is due to the subsequent effluent of inflammatory cells and cytokines, and it's marked by devastating tissular and vascular changes leading to local destruction. This secondary lesion is a major contributor to overall morbidity and mortality, and it represents the goal of several medical interventions, one of them being the pharmacological approach. The destruction of the Brown spinal cord barrier marks the start of this secondary phase that continues to evolve over time. Local microglia is the first immune cells to be activated. Then, other circulating immune cells infiltrate the spine and release their cytokines. The inflammatory process takes place, and finally, the glial scar will form. The damage is essentially due to pro-inflammatory molecules, free radical overload, glutamate excitotoxicity, hemorrhage, hypoxia, and edema formation, which will be the target of this project. So edema formation is one of the earliest events after injury. It increases the intraspinal pressure, which compresses the spare neuron, causing more necrosis. These events represent an endless loop leading with over-inflammatory process to massive cell death. Hence, the importance of early intervention in order to break these chains of events. The classical strategies used nowadays to manage edema are not efficient and act on reducing edema, once developed instead on inhibiting the underlying mechanism. In this project, we hypothesize that deactivating mast cells and blocking aquaporine water channel may reduce edema after spinal lesion. So let's first start with the mast cells. Recent works have shown that their role is not limited to allergic reaction. Their importance in inflammatory cascade after injuries is well revealed. In fact, they play a dual role. In early phase, they promote edema formation after degranulation and histamine release, while in a later phase, they eliminate cellular debris. The hypothesis is then to test the role of the chromoline sodium, a mast cell stabilizer approved for human use, in avoiding early effects and edema formation. The second hypothesis is about water channel, known as aquaporine. The aquaporine 4 is the principal subtype in the central nervous system. Blocking this molecule in order to limit water influx was until recently hypothetical for lack of such molecular agent. The trifluoperazine, an antipsychotic drug approved for human use, has been recently discovered to act on aquaporine deactivation by internalizing and delocalizing them from cellular membrane. So to test these two hypotheses, Mostafa is doing animal experimentation on rats. He makes thoracic contusion on the thoracic vertebral 10, so T10, in order to obtain lower limb paralysis. He does so by using an impactor connected by a specific device. He reshapes the impactor form to generate reproducible injuries. Furthermore, he made the model more accurate to study edema effect by adding 3D printed plates to recover the injured spine in order to conserve the intraspinal pressure, mimicking the real-world scenario. By using a classical method, he described for the first time, to his knowledge, the kinetic of edema evolution after spinal lesion. He found two peaks, one at 5 hours and one at 72 hours after the injury. As known, there are two types of edema: vasogenic edema, which is extracellular water accumulation in the tissue, and cytotoxic edema, which is the intracellular water accumulation. These two types are largely interdependent. It was not clear if, due to their interdependence, the onset of the second wave overlap with the onset of the first wave or not. So Mostafa made a predictive model to show that the two waves begin at the same time early after injury. This prediction was important to define the pharmacological approach. So in the light of this, the protocol was simply to inject these two molecules as early as 2 hours after the injury and see the effect on the edema evolution. First results indicate that none of these molecules reduce the first edema wave. Nevertheless, both of them can reduce significantly the second wave. And to see the long-term effect of this reduction, other group of animals were kept for 3 months. The sensorimotor performance rate of the rates were analyzed weekly by a set of behavioral tests. Then the recovery was evaluated after 3 months by electrophysiological tests like Hoffman reflex and evoked potential. Before euthanasia, spinal cord were conserved for astrological assays. So now Mostafa looks forward to see. So the aim is to discriminate between the cytotoxic and vasogenic edema by MRI using apparent diffusion coefficient. This approach enables him to make a link between the treatments and the specific type of targeted edema. Thanks for your attention. And now, if you have questions, we'll be pleased to answer.
Yeah, thanks everyone. So if you have questions, you can ask them just by raising your hand or ask them in the Q&A section or in the chat, but it's easier in the Q&A section. So we have one from Rochelle Acarlet. It's written Mostafa, but it's for Guillaume. Very nice detailed work, thank you. I was wondering, what do you expect to see at 70? Will it just give a better signal, or do you think you will be able to see more detail?
So we expect to see, so I did simulation with the relaxation time of the tissues. We expect to have 10% more signal, and with the gain of signal to noise ratio, we expect also to increase a bit the spatial resolution. So we expect to gain on both, on the signal and also to see more details. But it has never been done with the sequence, so we will see.
Okay, thank you. We have one question from Pascal. I think it's for Guillaume too. Can you evaluate the strength of the mechanical constraints, and can you correlate it with the severity of the disease?
So this work will be, I will do this work on Montreal. So I expect to see a correlation with the simulation and what I'm seeing now with the perfusion map. But I first have to improve the post-processing of the data to have better perfusion map, and then I will do a biomechanics simulation, and I would like to see if there is a correlation between. If it's,
Okay, thank you. Maybe a question for Mostafa. Is someone have a question? Do not hesitate to raise your hand and to ask your question directly. No. Okay, Mostafa, you are lucky. So you can always ask your question in the Q&A section and Mostafa can answer it. Ah, we have one hand. Let's see.
Can you hear me?
Yeah.
Hi, Mostafa. It's Anne Carmoudias. Do you expect, so as far as I understood, you expect a better recovery after edema reduction? But is it the only expectation you can have, or because I guess that as you said, the inflammatory has a benefit part as well. It's so do you think you will get only a better recovery or some other secondary effect, maybe unexpected one?
Hello, thank you for the question. I think that I'm waiting for only for beneficial effect, because
okay.
For me, the mast cells and the aquaporins have dual roles. So at the first time, mast cells have a role in the developing of edema, but there are dilated role in the phagocytosis of the cellular debris. So I'm deactivating mast cell just in to deactivate the first role and not the second one.
Okay.
So I'm waiting just for the beneficial effects. And for the aquaporine, the same, because aquaporine play a role for the to reduce edema in the second time. So I'm just deactivating them or delocalizing them just for the first part, not for the second.
Okay.
Okay, thank you, Han. Maybe the last one from Olivier. What do we expect from these treatments after SCE? SCI. Sorry.
Sorry, I didn't get the question.
You can read it if you can see it in the Q&A section. What can we expect from these treatments after SCI?
So we can have more spared neurons, so less of neuron death and better recovery.
Okay, thank you. And the last one from Pascal.
Hello, thank you. Thank you both for your nice presentation. I have a comment. In fact, I mean, both your thesis projects are really close. And I have now a question for Mostafa. I mean, do you think in your model of spinal cord injury, and do you think you could tackle this question and maybe find a way to, link your two projects?
Yes, actually, especially for the vascularization, because mast cell and histamine is not so far from that subject, but I have had some discussion with Guillaume about that, and it's a nice collaboration. It's a nice subject for collaboration, maybe after the thesis.
Okay, great. Thank you.
Thank you.
So thanks, everyone. So we are gonna welcome Maxime Cazorla and Christelle Bonez. And Christelle Bonez will present Maxime work. I will just share my screen with this. Okay. Christelle, you can go.
Yes, okay. Thanks, Ingrid, for asking me to invite, in fact, Maxime Cazorla, who was recently joined Marseille and especially the INT. to co-direct a team with Eduardo Gascon, the Sonic team in our institute. But Maxime is also very much involved in the development of the Neurotech Center for the next INT in its third version. Very exciting topics he's going to present today. But before he did a PhD in neuroscience at the University of Paris 6, as is written on the screen, in 2008. And then he did the first postdoc at Columbia University, in which he worked actually on the network of the basal ganglia, which is something in Marseille we are many people very much interested in. And then came back in France on a position in the Institut Curie in Orsay. And he specializes in the development of innovative technologies, as I said, in order to manipulate the architecture of healthy or diseased adult brain circuits. And today he's going to speak in particular about the photoinducible tool. So he's combining photoinducible molecules and brain-on-a-chip platforms that is using to rewire neuronal networks in vitro on demand. So you will see from what he's going to present that this very stimulating and innovative work as applications for the study of brain plasticity. So that it's in direct link with what we've heard before on the spinal cord injury, for example, and for the development of optoelectrical strategies for medical interventions. So we are really pleased to have Maxime actually within the community of neuroscientists in Marseille, and to have him as a speaker today. And I give him the floor.
So, okay, does that work? Do you see the screen? Yes?
Yes.
Okay. Thank you very much, Christelle. Very nice introduction. I'm also very happy to be here myself in this very rich community of neuroscience in Marseille. So today, as Christelle mentioned, I'm going to talk about the project that we have been working on lately in the lab, where we try to develop a new technology, which is a functional photoinducible tool that would allow us to manipulate brain connections artificially. This is what we call the manipulation on demand. And probably the first question to ask here is, why do we need to manipulate brain connections? And actually, this project is based on observations that I have made during my postdoc at Columbia. And as Christelle said again, I was working in the field of striatal circuits in basal ganglia. And especially what you can see here, so I was, for instance, do you see my mouse? The mouse is working, right?
Yes.
Yes.
Perfect. So in particular, so this pathway called the direct pathway, where medium spiny, a subpopulation of medium spiny neurons from the striatum is projecting directly to the substantia nigra pars reticulata. And you can see that they have these very, very long projections, and they pass through an intermediary nucleus called the GPE, globus pallidus external capsule. And this is what you can see here on this tracing, where you see that the axons, the projections are just crossing, and they make very, very few, almost no collaterals, no connections to the GPE. And now, if you inject channel rhodopsin, you use optogenetics, you implant fiber optics in the striatum, and you specifically activate the direct pathway. This is what you see on the video. You see that you can trigger locomotion, you stimulate locomotion in this mice. You can even see here, you can even tell that this mouse was implanted only on one side. So this is why you have this contralateral movement. So it's a very robust model to study motor control. And I won't go into the details, but basically we were working with a mouse model, a transgenic mouse model, and we made this observation that in this mouse model, these direct pathway neurons, they were making intermediary connections to this GPE. So this is something that you can see here. Now you see that you have a massive sprouting of collaterals here in this GPE. So now these connections, so first, these projections, we showed that they make functional connections to the GPE neurons. And now if you repeat the same stimulation, the same channel rhodopsin experiment, and now you stimulate direct pathway neuron, while these neurons are connecting both the SNR and the GPE, which is the indirect pathway, this is what you see here on the video. Same manipulation, but now the outcome is very, very much different. The mouse is struggling between a go and a no-go signal for the same stimulation. The same neurons are now being rewired and transmitting different messages from the original message here in the control mouse. So you see that now the mouse is very, very much, the behavior of the mouse is very much different. And this led us to the conclusions that actually rewiring striatal circuits in the basal ganglia was interesting to control motor behavior, the motor balance of mice, particularly in mice. So that was the, that was the basis of the project that I'm talking about, because at the same time, in these years, a new concept was emerging, and this is called the brain rewiring concept. This concept is very simple to understand, is that the adult brain is capable of extensive rewiring during learning and during specific types of learning. So let me go through this. So this is a naive control situation here where you have different circuits that we call here modules. So they are preformed, they exist, and they are believed to be associated to different types of processing for information, different types of behavior, etc. And when you do, let's call it a typical learning, you go through, you see a synaptic plasticity, what we would call in this case the micro circuit rewiring, where connections get potentiated or depressed, and that would lead to the weakening or strengthening of existing circuits. And now this would be associated with memory formation, okay, or the learning of a simple task. But some studies, and in particular in the non-human primates, they have shown that actually if you go through extensive learning to learn a complex motor task or complex visual task, they show that actually the brain can undergo axonal rewiring, so this massive macrocircuit plasticity. And they showed that, so it's here summarized in one very simple figure, but of course it was in different studies. So they showed that some existing modules were now capable of bridge other modules, or even existing circuits were disconnecting from a known target population. And now you understand that if you go through this extensive rewiring, now you completely change the module, you completely change the organization of the circuits. And this is associated, this is believed to be associated to the learning of some skills or complex tasks. The problem here with these studies is that they are all based on indirect observations. So there is no direct link, direct causality between the rewiring and the acquisition of a new skill. So this is why this problem led us to try to find a way to assess this direct causality between rewiring and behavior and learning. And that was how this project was, that was the birth of this project. And we came with an idea that we called RAIL. RAIL stands for Remodeling of Axons Induced by Light, where we would be able, in theory, in principle, we would be able to go from a known circuit to the creation of an artificial one that we could associate with a specific behavior or specific learning. So this is summarized here, very simple to picture. So this is a naive situation where you have an existing circuit. In this case, this is the cortical, very simple cortico-striatal circuit. And now, using these tools, in theory, we would be able to either create new connections or disconnect existing circuits to try and mimic what was observed during naturally during the extensive learning. So that's the basis of the project now. How we choose to do that? So does the rail technology looks like? So basically, it's based on the bioengineering of photoinducible activator of cytoskeleton dynamics. Again, very simple drawing here. So we have an effector that is trapped away from its target. And the cager is called love trap. Love trap is a protein that exists and that is sensitive to light. And this allows us to cage an effector and prevent its interaction with its target in the dark mode. So this is the control situation. Now you shed light on this construct here, and the love trap will unwind and release the effector. Now the effector can bind to its target. And then, depending on the couple you choose, the couple between the effector and the target, you would know, in theory, you're able to create either the rail minus, so for disconnection, so let's say for the pruning of existing branches, the collapse of existing connections, or the opposite, what we call the rail plus, which would be promoting the growth of new branches or the extension of existing branches. So how do we do that? Up here, how we do that. So those are the prototype models of the plasmids we're working on. So you see, we have different elements here. Of course, we want it to be specific for neurons, so we have a specific promoter. We want it to be enriched in axons because we want to remodel axons. So we have this axon targeting domain fused to a fluorescent tracer, M-cherry in this case, and all of that is fused to the love trap effector couple, which is the photoinducible effector. And we locked it so that we can have a spatial and temporal control of the rail. And actually, the idea on the long-term project is to associate it to a second virus, which is based on the ESER promoter that has been published very recently, which allows the expression of fluxed construct induced by activity. And this is particularly important for us because you can really imagine that if you start to change the connectivity pattern of any neurons, you would get probably a bad outcome. So the idea is to target only the neurons that are engaged in the task, that are activated, that are important for the learning. So this is why we chose to have this strategy. But again, it will be on the long term. We are just preparing this construct already. Okay, so how to test rail? Of course, the idea of the long-term project is to target non-human primates to try and repeat the seminal experiments that I've talked about in the introduction. But as you know, it's not an easy task. So we chose to go through an intermediary system, much simpler system, the mouse in this case. And the problem is that, again, to screen for the different effectors and to try to have to test different candidates, it's very difficult. I didn't mention it, but we have identified almost 40 of these candidates. We are working on 8 at the present, and we already have 4 of them to test. So it's a very long work. So of course, even the rodent becomes very complex in this situation here. So the idea is to go again on a much more simple system, in vitro system that, and this is why we chose this brain on a chip approach. So what is a brain on a chip system? So brain on chip system is a very big word for compartmentalized neuronal cell cultures. So basically it's cell cultures in vitro that you grow on this little microfluidic thing on that integrate a matrix of microelectrode array. And as you can see here, so in this case, for instance, the idea is to reconstitute the corticostriatal system and try and mimic the physiological architecture of the corticostriatal system. So here you see cortical neurons in the presynaptic chamber, and you have striatal neurons in the postsynaptic chamber. And you can see that these neurons, this population, they are separated by different types of microchannels. And in this configuration, we have axons from the cortex that project down to the striatum, but the striatal neurons, they don't project back to the cortical compartment, which would be unphysiological. So for that, we use different physical and chemical constraints. I'm not going into the details, but basically we have this configuration here where every element is identified and isolated. We have the presynaptic compartment, the axonal compartment, the synaptic compartment where cortical axons contact specifically striatal dendrites, and now you have the postsynaptic element. And you see that we have electrodes in the presynaptic electrodes, postsynaptic electrodes. So it's very interesting for us to stimulate or record this network here. So why this brain on a chip is interesting for us, because as I said, everything is compartmentalized, everything is separated and very well identified. So it's very easy to study whatever we want. And one of the first things we want to see is axon branching, of course, with the rail. So here it's a good example of differences that you can capture in this condition where you have only cortical axons in the striatal chamber, and you can check whether you see changes in terms of branchings before and after illumination. You can also study synaptic contacts, either at the structural level, using appropriate markers, or at the functional level using reporters. In this case, this is a reporter that senses glutamate that would be released by the cortex to the striatum. So you see spines here on dendritic striatal dendrites on spines. And here you can also study what's going on the dynamics of axons, in particular trafficking or cytoskeleton dynamics, remodeling. So in this case, I show you mitochondria trafficking. So you see it's very easy to study the kinetics of trafficking in these conditions. And of course, you can also study at the population level how here the striatum responds to cortical stimulation in different conditions using GCAM-CSF and calcium imaging. So we have a very nice tool that is very convenient to study the different elements of the network at the subcellular level, but also at the population level. So now we are in the position where we can screen our candidate and we can see what construct is the best. So we transduce cortical neurons with our different rail system. We illuminate specifically the very distant part of the axon projections, and we try to see if eliminating will rearrange rewire the circuits. Of course, I forgot to mention that this is not in a, let's say, developmental mode, but more on a mature mode. So we wait for the culture to be established, mature. So all of the experiments, all the photoinduction that we do are, of course, beyond 14 days in vitro to try and eliminate any developmental effects here. So here is an example of what we see so far. So you see 3-hour elimination of this region here, and we can see that we're able to promote the growth of branches in the middle of en passant axons here. So we have examples here, we have an example here and here, and you see that it's very dynamic, but the existing axons, they don't change so much. Nothing happens to them. It's mostly collateral branches that emerge. And we also have this very interesting example here where actually the culture was very old, dying, and you can say, as you can tell, the axons were pruning spontaneously. And we said, oh, okay, let's try to eliminate this region and see what happens. Can we regrow axons, basically, even if they are dying? So it's a longer stimulation process with a picture every 5 minutes. And this is what you see, so dying of axons, and now you see that the eliminated area is becoming populated again by neighboring axons that get stimulated and that will now stay or reach this eliminated area just because they have the RAIL plus construct here. But as you can say, probably from this video, it's cool for us. It was a very, very good first step. But the problem is that we have no control of it whatsoever. You see that some grow, some don't. Here we have an attempt, but nothing stays. And in terms of directionality, it's even worse. So we have no control of the directionality of these axons. They grow wherever they want. Here it's a pretty obvious case where axons come from above, from the side. They make a U-turn, they do whatever they want. So we have absolutely no control of the directionality and no control of the connectivity. So far, we are not able to tell them to stop growing and stay here and try to connect the target. So this is the second step. This is what we are currently working on. And for that, we chose another type of control in collaboration with Rod O'Connor from École des Mines in Gardanne, and who is joining us to develop the Neurotech Center at La Timone. And so the idea here is to use a new design in which we incorporate a new set of microelectrodes and to play with a phenomenon which is known as galvanotaxis, and which postulates that neurons are attracted by cathodes. So basically it means that neurons are spontaneously attracted and directed toward the cathode when you play with very, very weak direct current electric field. So the idea here is to create some kind of electrical tunnel to embed growing axons and tell them you grow in this direction because the direct field is in this direction. So the idea would be to attract them toward this direction, then change the field and say, no, you make a turn on the right, and then you grow again in this direction. So that's the theory. And when we will have tested that, the idea would be to combine optical and electrical stimulation to stimulate both the rail induction of the growth or collapse, and also to have now a good control of directionality and supposedly of the connectivity by stimulating the postsynaptic targets here. All right, so of course, so now we are in vitro, working with our brain-on-chip systems. The idea is to go as fast as possible into rodents to test this idea in vivo, sorry. Of course, there will be probably a lot of refining, a lot of problem in vivo, so we'll have to test different solutions back to the in vitro system and make loop. So one day we will be able to play with non-human primates in an attempt to recapitulate what has been shown naturally in the very complex task, motor task and visual task from the previous papers I mentioned. And then we will be able to have this link, this direct causality link between rewiring and the learning of a new task. And lastly, of course, I just wanted to talk about future applications. So, of course, as I said, there is a direct applications in connectomics to better understand brain circuits and their plasticity, the brain rewiring concept by playing directly with the architecture of the brain. But there is also a very interesting therapeutic interest here, I think, by developing these innovative optoelectrical strategies in an attempt to restore broken circuits in neurodegenerative disorders, but also in lesions, as we have seen from the two previous talks here, where you have a lesion somewhere, you have a disconnectivity somewhere, and the idea would be to try and reboost the regrowth of the axons that remain. A good example would be lesions, spinal cord lesions, of course. Another would be Parkinson's disease, where you would still have some projection left. And the idea would be to boost their reconnection there to try and compensate the loss during the degeneration. So, of course, it's a very long-term project, but that's the idea on the long-term goal. So with that, I would like to thank all the people who contributed to this project. Of course, the team here at INT, my past colleagues at Grenoble, at the GENE in Grenoble, our current collaborators in Montpellier, École des Mines and AMU, and of course the facilities on INT, INP, who allows us to do many things, and of course the funding agencies for obviously funding all of these projects. And with that, thank you everybody, and if you have questions, we'd be happy to answer it. Thank you.
Thanks a lot, Maxime. Oh, sorry, Christelle, you can go if you want. So you can ask your question by raising your hand or in the Q&A section.
Waiting for a question. Can I ask a first one? I wonder, in the model of the corticostriatal brain on chip that you have initiated, at the stage of development of the cells you use for this model, can you already have the functional specialization? Because we know there are loops, the motor loop, the associative loop, and the limbic one. So depending on which cortical territory you would use, do you think you would find this functional specialization? And if so, is it possible to think that you could model the spiral model described by Susan Haber just on a chip? Would it be possible?
I want to say yes and no. Yes, to be optimistic, and no, to be realistic. The no is obvious. It's just because I'm using progenitors here. So I take the, let's say, all cortex, embryonic cortex, and striatum. So of course there is no functional territory here in what I use. Now, it doesn't prevent us from creating fake loops. So basically, you can design whatever you want. So you would just use cortical neurons, striatal neurons, all the same. But then you would try to create a design in which you create this loop. So you would force the cortex to talk to the striatum, then the striatum to talk, let's say, to the thalamus, and then back to the cortex. And then you make a second chamber where it goes to another striatum. So you can do that. There is no problem in doing so. So this is the yes, this is the optimistic version of the answer. But of course, just like that, no, it's not possible.
Maybe Pascal Jorbet, I can follow up on this point. This was a little bit my question. I was wondering if you can use this brain on chip system to have a more complex, mixed of cell population, for example, obviously including oligodendrocyte cells or microglia cells, just to check how these cells can, be part of the connectivity system, and obviously it would be really interesting with microglia also.
That's an excellent point. Yes, of course, oligodendrocyte and astrocyte is already something that we have thought of. Of course, we cannot do everything at the same time, but oligodendrocyte is very, very important here, mostly for the rewiring, because myelineated axons are very unlikely to rewire, so we'll have to study this effect in vivo. Most of the axons are myelinated, so those are very important questions. Now, so we can complexify the system as much as we want, as long as physically this is possible. So this is not a problem. Now we always have to take into account that this is a simple in vitro system. So sometimes we try to complexify it just to mimic the physiology, but then it doesn't make a physiological, it doesn't answer a physiological question. There is still a limit. It will still remain cultures.
Yeah. I had a second point. There is something which was not clear for me in the electrical stimulation system. Do you have only one cell population, or can you combine both of them?
No. So the picture is just a matrix of electrodes without the actual chamber on top of it. There will be a physical constraints here, compartmentalization, depending on what we want to study. So basically, it will be the same. So we will tell cortex, connect this axon, and then now the axons that are here, those are the axons that we will direct specifically with a new set of electrodes.
So basically, you will try to make the axons have a new direction under the influence of the electrical field in presence of striatum.
Exactly. Yes.
Have you think about using, guidance molecules?
Of course, I did. This is something that I get most of the time. I do not want that, just because I try to make something global, applicable to any kind of circuits. And if I start to choose guidance molecules, the problem would be that I become specific to one type of circuits. So if I want to study this circuit, I would need to use semaphore in 3A, for instance, but if it's this one, I need to combine it with something else. So that would be very difficult for us to have a standard tool. So
thank you, Maxime. It's great to have you around in Marseille.
Thank you.
And we have a question from Clément. Clément, normally you can speak.
Yes, can you hear me?
Yeah.
Yeah, thanks a lot, Maxime. It was a fantastic talk. I'm Clément from INMED in Lumini. I'm sorry, maybe I missed it, but I have a first question is, what effector do you use in your rail plus and rail minus? I mean, maybe you don't want to actually say the molecules used, but
well, it's not super secret, but we have different candidates. So it's either we have the traditional raw GTPase effector, RAC, ROA, etc., CDC, for instance. But we also have other candidates that would regulate, for instance, mitochondria trafficking or metabolism that could be used either independently, just to see if they can have a role. So in this case, if it's mitochondria, for instance, it would be very, very independent on cytoskeleton dynamics. But we are most likely thinking that it will be useful as a combination. So maybe we'll have to combine two different candidates targeting different processes. One targeting cytoskeleton, actin remodeling, microtubule initiation, and another one bringing, for instance, energy with mitochondria trafficking, for instance. So we have a very long list based on our previous work, but also, of course, on the literature.
Okay, and thanks a lot for that. And did you look for functional synapses? Did you check that?
Not yet. So this is the very step that we will do now. actually. We will actually use first GCamp, as I showed, the GCamp analysis, just to see if we have changed the strength of the transmission just by applying the same stimulation before and after rail. We will use igloo sniffer and so on. So for now, we haven't done that. It's just anatomical, let's say, just structural for now.
Cool. Thanks a lot.
Thank you.
And we have Lydia. Normally you can speak, Lydia.
Yes, thank you. Thank you very much, Maxime, for this very exciting project. I was wondering, don't you think that changing the connection, for example, from the cortex to the striatum, will also change as a result the connection, for example, between the striatal neurons? And is this one aspect you will investigate?
Yes, that's again an excellent point. Yes, of course, we are expecting that, and that will be so. When we will have reached this situation, I would be the happiest because it means that it works and something is happening. And if we can correlate that to a specific behavior, I would be very happy. Now, studying what would be the downstream effect of this rewiring and comparing to the natural situation where you train mice to learn a specific task, and you compare to an untrained mouse that has been stimulated with these tools, and then we will see if we have differences in the behavior, but also in the local rearrangement between very locally and maybe also in the loop. It's very likely that there will be big changes in the loop. So it will be a timeline that we will have to study, different structural, functional rearrangement that appears. But yes, I would be very excited when we have reached this point.
Thank you. And for instance, in the very first experiment you described, where you reinforce the connection of the direct pathway with the GPI, does this change the effect of manipulating the indirect pathway? Because you connect the direct pathways with the GPE, so does this change the impact of the indirect pathway?
So the answer is no. There is absolutely no effect on the indirect pathway. So if you stimulate the direct pathway, you get massive inhibition in the GPE. It is not changed after the operation of these collaterals. Behavior is not changed. So here there is absolutely no weight of these collaterals on indirect pathway. So we have seen indirect a loop effect actually on the corticostriatal projections that are probably changed because of this loop. I don't have the direct proof, so this is why I don't talk about it. I don't know if it's through the loop or very local effect for now. But we saw dramatic changes also from the corticostriatal loop. So probably there is, when you have, as we were mentioning, when you have a primary remodeling, then you have secondary, tertiary remodeling in the whole network, in the whole circuits there.
Thank you.
Thank you.
Thanks a lot. I think we don't have any more questions, or if anyone wants to ask it now. Okay, I think it's okay. We just share my screen up. Okay, so thanks everyone, and we are very warm to welcome everyone each time. So we will see you in January, but not with a webinar, but with a tripartite symposium. On January 21st, it's on all day, and we will welcome different researchers from the Sapienza, from Tübingen, and NeuroMarseille, of course. So do not hesitate to book your day, and more info will come in January. So thanks everyone and have a nice day. Oh, 2022, not 21. Sorry. Bye-bye.
All right, thank you. Bye-bye.